All right, everybody. We are Unit 5, Lecture 1. One thing that I know she's probably going to have you remember, is a guy named Mendel. Mendel, lovely handwriting. This is the father of genetics, okay? This is the guy who came up with it all. This is the father of genetics, and he basically did everything um, by playing around with pea plants. What part of a plant do you think is a zygote? What part of a plant do you think is a zygote? Pause the video real quick and see if you can think. Okay. The seed is going to be a zygote because plants have male and female parts and they pollinate and they create a zygote. However, some plants, like the ones that, like the pea flower specifically, that he studied to come up with all the four principles of genetics that we go by. This was a plant that had both of those parts together. Okay, so if it pollinates itself, if a plant pollinates itself, okay, this is known as monoecious. You're gonna have to know that. If it pollinates itself, basically it's one plant that has both male and female parts. Moving on, we're going to have to kind of define some things here. There's going to be a whole lot of letters, and you need to make sure you don't get these confused. So if we're talking about genetics, we're going to be talking about offspring, we're going to be talking about parents, children, grandchildren, great-great-grandchildren, and that can get really confusing. So we have a letter P, which, it's P generation. P generation means the first generation. Okay? Do not get confused thinking that this P means parent. You might be tempted to think that this means parent. Okay? It does not. It does not mean parent. It means the first generation. The very first. That way, instead of, whenever we're looking at maybe a long lineage, instead of saying the great, 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 great grandparents, we just say P-gen. Okay. Below that, we have F1. And this is the offspring of P-gen. F1. Okay. And guess what? This is going to go in order. F2, F3, until we reach whatever the number is, F of n. It does not matter. So I doubt she'll do this, but F34 is 34 generations away from P-gen. So you need to know P, what it means, and what it's going to mean when we talk about F1, F2, so on and so forth. Okay, so next, let's just go a little smaller. We have Mendel's 4 principles / laws. Principles, laws, same difference. Okay, and we're going to go over these. This is going to be different from lecture. You have to wait for all these to get them at once. The first one, now you know most of these in your head. That's the thing. Inheritance. Inheritance. And what this means is info passed from both parents. Info passed from both parents. The reason why this is important is that when Mr. Mendel was doing his genetic research, it was believed that no traits were from the mother and all the traits were from the father. The second principle is dominance. Dominance. Okay. This just means if you have a dominant form a dominant gene, it will be expressed. It'll be expressed. Next, segregation. Segregation. And guess what? You know these next two. Segregation just means that the cells that come together, that basically this means that the chromosomes separate. The chromosomes separate, like meiosis, right? We already know about meiosis. This is literally just talking about this. This means we have to have haploid cells. We already know how that works because of Unit 4. Guess what the fourth one is? Independent assortment. You probably can't read that, but independent assortment. So again, inheritance, dominance, segregation, and independent assortment. So this was kind of sloppy. So what I'm going to try to do is actually type the next ones out because it's really important what the next things are that I'm about to tell you. We need to know what a gene is. A gene is just any sequence in DNA for a specific either protein or trait. So it's any sequence of DNA. And where are the sequences of DNA? Remember how there's bundles of DNA and histones inside of a chromosome? And you have multiple genes inside of here. There are multiple genes in between these, right? So genes are linked to proteins and traits. Basically, a gene is, now trait, it already makes sense to you, but why protein? This wasn't really talked about in the lecture, but I think it's really important that you know. What inside of a cell is responsible for making proteins? The ribosomes. And what do ribosomes have in them? RNA or instructions. So a gene, other than traits, is an instruction on how to make the proteins. Now, a couple things that's important is you could have a gene, by the way, that is not expressed as a trait. So this is when we get down to traits. So right now, also, we're not going to go any further into proteins. So stop worrying about it. I just wanted you to know that little factoid. But she does not get into protein synthesis until the next lecture. So we're going to move on. Let's talk about this, okay? So if you have a trait for something, but it's not being expressed, right? So let's say I have a trait, I have a gene for blue hair, okay? Just something crazy. I could have a gene for blue hair, but I do not have blue hair. That means I have the gene and I don't express it. In other words, you can't see it. What is this called? This is actually the real definition of carrier. So carrier just means you have a gene, but it is not expressed, aka a secret gene. So talking about traits, there are two kinds. So you can see blue hair. If it's something that you can see, it is called phenotype. This is something you can see. You can see if I have blue hair or not. These are physical traits, physical expressions. Phenotype. The way you can remember is PH, think physical. Contrary to phenotype, we have genotype. And this is, a genotype is all genes, whether it is expressed or not. So this is all the genes that you have. Let's say that I have blue hair. Let's say I have blue hair now. Yay, this is me. I have blue hair. Let's say that I have blue hair, gene, and not blue hair gene, okay? let's say that I have a gene for green hair. I'll explain why these are two Bs in a minute. So this blue hair, this is my phenotype. And the fact that I have a gene for blue hair and a gene for green hair, this is my genotype. These are the genes that I have. But this isn't expressed because I have a dominant blue hair gene. So really quickly, let's get into why this is like that, okay? Let's keep going with blue and green because I think it's a little more out there. So capital letter is dominant and B is the first letter in blue. Now, whenever we talk about pairs, we're going to have a dominant, we're going to have two different genes, and one of them is going to be expressed, right? So we have, let's say we have this, and let's say we only have one other alternative. This is really important. Pay close attention. Let's say we can only have blue hair or green hair. Now, I know this isn't reality, but let's just say we can only have, but instead of putting a G here, we would just put a lowercase b because it's either going to be blue or it's going to be green. But we're going to have the letters be the same. It's just that the alternate is going to be recessive. It's going to be the same letter. It's going to be lowercase. So this lowercase b here is recessive. And I'm going to share with you why the hell that matters right now. So let's do something a little closer, okay? Let's do tall and short. Now, again, maybe it wasn't clear to you a second ago why we only have two. So just in case you're hung up on that, one comes from the father and one comes from the mother, right? We already know about that. That's how genetics work. We have pairs of chromosomes. So we have two different sets of genes, one from the mother and one from the father. So the combination of who has what and which ones are dominant or recessive ultimately controls your phenotype from the genotype. So I'm at the same time going to introduce you to the idea of Punnett squares. So we have tall or short. Now, again, this only works when, and this is how things are going to go for the test and the class. We only have two different options. This only works when we have two different options, and that's what we're going to be doing in this class because it's not a genetics class. So instead of writing an S here, again, we write a little t. This is just for not tall, which in this case means short. Now, if we go back to the rules here, dominance. If dominant gene is there, it will be expressed. This means that if there is ever an uppercase T, that person will be tall, right? If we have just tall and short. So the way that we do this in a Punnett square is we're going to divide this up like this. And we have one parent here. And let's say they're tall. And then we have a little t here. Okay? So this is the genes that the mother has. And this is the genes that the father has. Let's say we have another t, and let's say we have a little t. Now, this is all the information that we have to know. Why does it keep doing it? In order to tell us what is the percentage chance that we're going to have when these two people, whoever they are, when they get together, what are the chances that their child, that their offspring will be tall or short? So all you have to do is combine these. So here we have two T's, two uppercase T's. You have to be really careful about that. Right there at the top, so you don't get it confused with the lowercase t's, okay? Now, is this if this is the outcome, then the offspring will be tall because we have at least one capital T. Now here, this combination, we have a big T and we have a little t. Now, is this person going to, if this is the outcome and how the genes line up, will the offspring be tall or short? They will be tall because this T is here. This is recessive and this gets completely ignored. Now here we have this uppercase T and we have a lowercase t. So is this offspring going to be tall or short? Going to be tall. And lastly, if this is the combination, we have a lowercase t and we have a lowercase t. Is this person going to be tall or short? Right. This person is going to be short if this is how the genes happen. So in other words, you have a 25% chance, of being short. This is how the genetics problems, this is what we're looking to find out, is what is the likelihood the offspring will have X trait. That's all we're figuring out here, okay? Bada bing, bada boom. Again, this is a Punnett square. This is how we line them up. Bada bing, bada boom. Now, we have, with two genes, we only have a few different options with how these things work. We have T, uppercase, and we have another T. This, because these are both the same, as far as dominance is concerned, this is known as homozygous. Homozygous, because we have two dominant genes here. If, however, we have this is called heterozygous. And the same thing, if we had this, two lowercase t's, this would be what? This would be homozygous because they're both the same. These are terms that you have to know and understand what they are. These things are the same. These things are different. One's tall, one's short. So it's heterozygous, homozygous. These are both the same gene. So if you haven't noticed here, dominance basically means that you won't have the trait unless the recessive trait will only be expressed if both genes from both parents are both recessive. Otherwise, recessive gets completely ignored. Okay. One more really quick term that we have to know for some ungodly reason is that of a locus, L-O-C-U-S, and this is a physical location of a gene on a chromosome. Not sure why we need to know that, but it's in the lecture and we need to know it. A couple more terms here. We're going to start talking about genetic expression a little bit deeper. This is really important. We have to know what an autosome is. An autosome is a chromosome that does not determine gender. In other words, these are not the sex chromosomes, right? So in humans, we have 23 pairs, right? Well, the last pair are sex or gender determining genes, or excuse me, chromosomes, right? We have 44 autosomes. There's a really important concept we have to know. And these concepts is the last bit of the lecture. Autosomal dominance. So what is this? What does autosomal dominance mean? So first of all, we have two phenotypes per trait. Two phenotypes per trait, right? In other words, this is important. I make the distinction. By trait, I mean eye color, hair color, height, skin pigmentation, whatever. right? This means we have to have one possibility or the other. So eyes, and this she does stay in the lecture. Eyes, we actually only really you could say that we only have two phenotypes for that if you wanted to, because you either have a pigmented eye or you don't. Blue is no pigment, and all of the other colors have some kind of pigment, right? So basically, two possibilities of physical characteristics per trait. Now, for humans, there are not many like this. The only ones like this are widow's peak, hitchhiker's thumb, if you have freckles or not. There are a couple others, but these are the ones that she gives in the example, so who knows, they'll probably end up on a test question. We have another thing called incomplete dominance. Incomplete dominance. Incomplete dominance, we have two phenotypes per trait again. We have nothing that's dominant. Instead of having dominant genes, we blend together. Okay? So basically what we mean here is if we have, so this is probably most of our genes, right? So I previously mentioned tall and short. Obviously, we don't have just tall and short. We have humans that are all different sizes, right? This would be an example of that. So if you have somebody that's really tall, right, a gene for being really tall and really short, these combine to like average height. If you have something, in the case of plants, she does use this. If you have something for a red flower and a white flower, these combine and they'll be pink, right? They blend together instead of just one or the other previously. So for autosomal difference dominance, what we're saying is you're either going to have a widow's peak or not. You're either going to have hitchhiker's thumb or not. You're either going to have freckles or not. You don't have half of a widow's peak. You don't have half of a hitchhiker's thumb. For incomplete dominance, we blend these together. So dominance does not matter here because we're going to blend together the different genes and have blended phenotypes. The next thing is going to be something called codominance. Codominance? In codominance, neither allele is dominant. Both expression in pure form. What does this mean? What this means, this is very strange. Let's say we have a gene for blue hair color and the green hair color, right? Let's do it over here so it makes sense. What this means is not that you're going to have a combination or a blending together, right? So this would mean that blue and green blend together and you get some whatever the hell weird color that makes. Autosomal dominance means, and that's what we were talking about up here with dominance, this would mean that we would have blue hair if we were using autosomal dominance. But codominance means that we are going to have both blue and green hair simultaneously. Whether that means half your head is green and blue, whether that means every other strand will be green or blue, whatever that means. That means you do not blend and you do not have one or the other. You straight up have both of them, right? So another weird example would be like if you had a gene for long toes and then you had a gene for short toes. If we were talking about codominance, that would mean that like your big toe would be like really long and then your next toe would be like really little and it would be random, right? You wouldn't have like either long or short toes, or a blending, like a medium or a blending together, you would just have some that are short and some that are long. So both of them dominate, in other words. What we're going to talk about now is something called X-linked recessive. So what this means is this is on the X chromosome only. Anything on X gets expressed because it's not 2 genes per trait. This is because in males, we have an XY and the Y is shorter and they don't have a defense. What I mean by that, we have an X chromosome, right? And we have like a Y chromosome. That means you see how we know that X chromosomes are larger than the Y chromosomes. Now, chromosomes are the size that they are because there are specific genes that are in certain spots. So if there's a gene up here for hair color, there's not another gene right here on the Y chromosome. So males will default to whatever is on the X chromosome. This is why you look like or more like mommy. Okay? This is why this happens if you're a male. So this is why males look more like the mother. It's because you have that shorter Y chromosome, so you don't have anything matching. So for a certain aspect, there are a lot of genes that, regardless of whether they are recessive or dominant, they will get expressed. That's why we have X-linked recessive. So if you have some kind of super rare thing on the X chromosome and it's recessive and it's been, let's just say that it, because of that, it has been carried for like 5 generations because they only had girls, right? And then all of a sudden, someone has a male offspring. They don't have a quote-unquote defense against that because this is a shorter chromosome. And even though this is recessive, this is going to be expressed anyway.
The journey is usually the part that you remember anyways.
So looking at this, the way that she's going to write it, this X means dominant. An X like this means recessive. And Y, this is a male chromosome. So this is the notation that you're going to be required to know. What's important to know is that this N is actually going to be, you know how we've been doing the letters for whatever trait it is? This N will be replaced by the trait, right? So let's say in this case, for the next example, we'll have a D for deaf. D for deaf. Let's do a Punnett square real quick. So let's say the mother, right, having 2 X chromosomes, here she has a recessive trait for being deaf, and we have the father here. So we're going to figure out what are the chances of a deaf child. Here we have 2X. Here we're going to have X, XD. Here we're going to have XY. And here we're going to have X, D, Y. Now looking at this, we have a male and a female, right? We have, if you notice this, we have 2 outcomes where we're going to have XY chromosomes and 2 outcomes where we're going to have XX. So this, if we have just a male and a female coming together and we're just looking at X chromosomes and Y chromosomes without any trait, you can see that there's a 50-50% chance of male and female, right? You have 50%, half of the square is male and half of the square is female. This is where we have to be really careful. So first of all, before we go through that, let's make sure we understand this person would not be deaf. this person would not be deaf because this is recessive. Remember that this is recessive in this notation. Remember that this is dominant. Now, in the dominant, we don't see a gene anywhere here in indicating deafness, right? So the dominant gene will be expressed, meaning there's a 0% chance of this particular offspring, the female, being deaf. 0% chance of the female being deaf. Pay very close attention to what I just said. Now, if we look at the male, we have no deafness gene right here. And if you notice here, we have the X chromosome with recessive gene for deafness here. This male will be deaf. So the male has a 50% chance and the female has 0. Here's where we get Wilma specific. You're going to have a test question, right? And for the following Punnett squares, of which she's going to give you the gene, she won't write the square for you, chances are, and she shouldn't. She's going to say, what is the percent that the child or offspring, actually, will you use that? Offspring or F1, if this is P generation, what's the chances that F1 will be deaf? Pay close attention. Offspring/child, there is a 25% chance, because we're talking about all of the possibilities, right? We have a 25% chance of the offspring being deaf. Now, this is different than if she says, what percent chance of a male offspring to be deaf? That is 50%, because we're just talking about males. In other words, we're ignoring all of this. If she's saying what percentage of male offspring, what's the likelihood of being deaf? Obviously, we have 50%. Same goes the other direction. What is the percent that the female offspring will be deaf? That's 0. So she could ask us the same set of genes, the same Punnett square, not just these 3, but several different questions. Like, oh, what is the likelihood of the offspring being male? What's the likelihood of the offspring being female? What's the percentage chance of the offspring not being deaf? What's the percentage of the male not being deaf? There's all kinds of questions that she can ask. So you have to pay attention if she's talking about the entire offspring, which is the entire square, or she's talking about males, or she's talking about females. You know she's going to try to trick you up on that. So pay really close attention. Now, moving on to the next concept. So we need to talk about cross types. We're going to talk about cross types. Now, we have something called a mono. This is not really that difficult. Monohybrid cross. Okay, and this just means a cross between 2 individuals, and we're following one trait. What do I mean by that? That means we're talking about 2 individuals, and we're talking about one trait. In other words, we're going to make a square, and we're only worried about either hair color, eye color. For example, we're only worried about trying to find out what the likelihoods are of that one trait being one or the other. And then we have a dihybrid cross, right? And I'm sure you can guess what this is. This is a cross between 2 individuals, but we're following 2 traits. So how many squares for monohybrid cross? 1, 2, 3, 4. So here we have 4 different possibilities, right? Well, if we're following 2 different traits, we're going to have 8. 8 in the Punnett square. In other words, we're following hair color and freckles. The last thing that we need to talk about is pedigree. A pedigree chart is going to look just like this. And this is really cool because we basically have certain genes, and then we also have the phenotype for each parent. So this is basically a little puzzle where we can use the offspring to figure out which gene is dominant and which gene is recessive, right? Because we know how dominant genes and recessive genes behave. And so if we ever run into an instance ever where we have 2 carriers and any one of the offspring is affected, that means that this must be recessive, right? Because we know, for instance, hemophiliac, again, I'm not really analyzing right this second, but hemophiliac, the only way that 2 parents that do not have the gene expressed, but they can have a child with the gene expressed, is if it is both recessive, meaning that that gene has to be recessive. So that's really what you're looking for here. But if you can figure out which gene is recessive or dominant, then you can keep going down the line of the pedigree, and you can figure out the chances that a certain offspring down the road is going to have a given phenotype or not, or even if they're going to carry that gene or not. The last thing that I'll say is there were a couple things that I needed to clarify that I didn't really kind of go into, and that is the definition of an allele. This is a version of a gene. In other words, if we have blue eye and brown eye, this is an allele and this is an allele because it's the same trait, it's the same gene, but different alleles because one will yield you blue eyes and one will yield you brown eyes. The second thing that I'd like to say is that one thing that you can posit from this male and female business here is that, and this is a really important thing even if it's not obvious, is that males cannot carry. Because of this, it is expressed. If they have the gene, it is expressed because of that shortening, and that's kind of really important to know on that. So males cannot carry for the sake whenever we're talking about X-linked recessive genes. A male cannot carry one of the genes over from a female that's recessive because they're going to express that gene. So they would not be a carrier, right? Because remember, a carrier means you have the gene, but it's not being expressed in your phenotype. So it's just a really important quick thing. I think that's it for anything that's required. We're going to do a couple examples of Punnett squares. Actually, I'm going to post that in a separate video.